And yeah, this is going to be a shambles. But anyway, I'm Julianne. I'm a master's graduate from Portsmouth, and I study prehistoric plants and basically how to restore them. And yeah, today I'm going to be talking about restoring the Bennettitales, which are a group. Well, yeah, they're a group of dead plants. Anyway, yeah. So yeah, first of all, just a quick introduction. What are the Bennettitales? So they are. You've probably heard the name. You probably know them as very cycad-like plants, which are very, very common in Paleoart. But yeah, they are incredibly common in the fossil record. We have like many, many genera of leaves and reproductive organs and everything, stems, scale leaves, all of these different things. And yeah, their leaves were mostly pinnate. They were very cycad-like in that respect, but their reproductive organs were remarkably flower-like and have often throughout the literature rather confusingly been called flowers. And because of this sort of dichotomy between their cycad-like foliage and their flower-like organs, we don't really know where they fit in the tree, the literal tree of life. They have often been considered very close to the base of angiosperms, but more recent phylogenies have put them closer to, or even within conifers, actually. So they were probably most closely related to modern gymnosperms in some way, but we don't really know. The interrelationships within the family are not really well understood. Mostly we have isolated organs, just leaf here and flower, well, flower in quotation marks there. But they are traditionally divided into two main groups. We have the Cycadioidea, which are, here's a cone of one here, whoops. We have Cycadioidea, which is composed of Cycadioideaceae, which is composed of Cycadioidea and Monthesia, just the two genera, and those are the ones which you see have the very, they basically look like pineapples. So the ones you see in Paleoart sitting in the desert, little and round with pineapple, with leaves coming out the top. And the rest of the organs we know of fit into a family which was probably paraphyletic called Williamsoniaceae, and that's the family I'm going to be focusing on today. I do not have enough time even for that. So anyway, yeah, I'll be focusing on Williamsoniaceae. So just a quick introduction, just a quick overview of the leaves. As I said before, they can be very cycad-like Zamites on the right-hand side. Their leaves were mostly pinnate, which means basically they had a long central stem with many small leaflets coming off the sides. And that is what you see typically in Paleoart. But there was a lot of variety, diversity of shape in the family, as you can see here. Like, all of these leaves come from various Bennettitalians, and we have various shapes, like Eoginkwites is particularly unusual. It basically looked like a ginkgo leaf. So much so that, yeah, hence the name. It was originally thought to be related to ginkgo. But we even have more unusual leaves like Nippo patellophyllum, which were bipinnate. So basically composed of, again, long central stem, but each one of the leaflets, instead of being just a single leaf, basically, they were individual fronds. And there are a couple of species like this, but they're not particularly well known, and as far as I'm aware, they've never actually been restored in Paleoart, which would be very interesting to see. And yeah, then again, we also have things like Nilsoniopterus, which were basically just had long strap-like leaves, sometimes with incisions in the sides. But one thing I found very interesting about the leaves is that lots of them have very dense leaflets, to the point where they basically create a pseudo-leaf. So if you look at things like Otozamites, the leaflets all overlap each other, basically creating a continuous surface that from a distance would just look like a long strap-like serrated leaf. Yeah, quick view of the reproductive organs. As I said before, they had very flower-like organs, but I'm going to try and avoid calling them flowers because that's very confusing and because they weren't flowers. But yeah, they were mostly... most of the plants were dioecious, which means they basically had male and female organs on separate plants. And yeah, there are some of the genera. The most important ones probably are Williamsonia, of which there are like tens of species. I do not know how many, but there are an awful lot of species. And they are always associated with, well, they're mostly associated with organs called Waldrichia, which are again very flower-like. They have often quite a deep central cup with resinous bodies, or basically nectar-producing organs, with spore-producing microsporangia on these radial lobes. And yeah, these two different genera are basically the male and female organs of the same type of plant. There are a couple of bisexual organs, and they are mainly from either Cycadioidea or Cycadioideaceae, or two other plants called Williamsoniella and one which is unnamed called the Lunds Bennet. Well, I've just called it that. It doesn't really have a name. So we don't really know how these are all related to each other, but these probably clustered together in a separate family or subclade, basically. And yeah. So this is just showing you the diversity of Waldrichia species, and this is thanks to a paper by Pepper in 2019. He basically just went and reviewed all of the known species at the time and did some nice restorations of them, which I have literally just coloured here. But as you can see, they were incredibly flower-like. We don't know what colours they would have been, but it's reasonable to presume that they would have been meant for attracting insect animal pollinators. But I'll talk about that a bit more later. So the main section I want to talk about is go over a couple of taxa, which I have basically looked at, restored, and basically just to give an overview of what we know of these plants as living organisms. But just a quick note, I've called them biological or whole plant taxa because unlike in vertebrates, you might be used to, say, we find one bone, we call it one thing, we find another bone, we call it another thing. Then if we find they're from the same animal, we call it the original name. In plants, it's slightly different because it's better to think of plants as colonies of different organs. So in paleobotany, a leaf will get given one binomial name, reproductive organ will be given a second binomial name, a stem will get another name. But when we find those organs are associated together, aka they would have existed on the same actual plant, a biological taxa, we often give that either a separate name or we name it after the female organ, or sometimes the first thing which was named. It really varies. But whenever we find the organs separate in the fossil record, which is how we mostly find them, we will still call it the original name. So unlike when we synonymize things in animal paleontology, we don't really synonymize things in the same way in paleobotany. Anyway, so the first one I want to talk about is Wielandiella angustifolia, which is the most well-studied of the ones I'm going to be talking about. It's known from Northern Europe and Greenland from the very end of the Triassic. And yeah, here are a couple of fossils. So this is the stereotypical Bennettitalian. This is the one which there was a beautiful illustration by Polyan von Noring, which has kind of shaped the way we draw Bennettitalians that shapes the way we've drawn them for the last 10 years. This plant was basically characterized by having these quite long, narrow stems which divided evenly. They dichotomized, is called divericate branching. Basically, they divide at a wide angle and then spread up and curve upwards. The stems themselves were quite narrow, and we know that they had these grooves in, so they would have had an interesting cross-section. And yeah, basically they continued to divide, and leaves were born in whorls at the apex of the youngest branches. And the cones were born again at the apex of these branches, so in the next season when they branched, they remained. An interesting thing about this taxa is that it seemed to show weird transitional leaves. So these are not known from many other taxa. We have standard pinnate leaves, but they also have leaves which are basically abract, which would have surrounded the cone, with little leaflets on top. So these may just basically be malformed leaves, but considering they're very common and are not known from other species, it's likely that they would have actually been present around the cone, basically encompassing it in the centre of the leaf clusters. As I said, we know a lot about this plant. We know everything from the way that it grew, basically the steps it would have taken. We don't know whether it would have been in years or whether it would be seasons, but it's a discrete unit of time, whatever that was. But yeah, basically, the stem grows, leaves grow, leaves drop off as the stem grows, and the cone forms at the apex, which is then pollinated within the first year. Then in the second year, the cone overwinters, and that is probably when the micropyle grows down to the seeds. So there is a delay before they start developing their seeds. And you get these little nodes which form underneath the cone, which then grow into the next order of branches. During the second year, these bracts would have basically just dried up and dropped off slowly, eventually exposing the cone. And then in the third year, the cone would either disintegrate or was potentially eaten by vertebrates, so it may have been a fruiting body, but I think it's more likely they disintegrated purely because we don't have abscission points. Basically, if they're eaten by an animal, there would have been a break at the very base, which we don't have.
Jules, I think you need to speed up a little bit.
Okie, that's great. Okay, anyway, so leaf clusters, they would have overlapped. This is quite different to what you normally see in Paleoart of Bennettitales. The leaves were longer than the internodes, so they would have actually overlapped with each other and interlocked. They would have created basically an open canopy obscuring the branches below. This is a cone development, which I basically just described. So cone is pollinated, cone grows, and the bracts drop off. And then basically it just all disintegrates. An interesting thing, we might potentially be able to tell some of their colour. So a lot of flowers have papillate cells which are filled with anthocyanins, which create like pinks, reds, oranges, those kinds of colours. And they're concentrated in these cells because as the light enters them, they're basically refracted out and they become really bright. We have papillate cells clustered around the heads of the interseminal scales. So it suggests that the scales of these cones were probably flecked with little bright spots of red or pink or orange or some colour like that. So that was very interesting to find out. Yeah, I didn't talk about the male organ because we don't actually have the male organ for this plant. But this was once thought to be it, but it's now known that Welsbergia was associated with pterophyllum leaves, which, again, is just an interesting thing. It was a different, closely related plant with a male organ which seems to be wind-pollinated, so wouldn't have had insect interactions. Next one. Oh god, I need to speed up. Weelandiella villosa. It's the same genus as the previous plant I was talking about, but it's a different species from China. As you can see, it would have grown slightly differently. The stems were narrower, they were straighter, and while they were about the same length as the previous species, or a little shorter, the leaves were potentially shorter than the internodes, which means that the canopy of this thing would have been very open. We also have developing buds in these dichotomies, so that could indicate that the specimens we have are young plants which haven't matured yet and don't produce cone, or it might mean that cones only developed in the second year, unlike in the first year of the first species. Here's just a quick comparison. As you can see, it was much smaller. with smaller leaves, but adult leaves did get larger. So we do have leaves which were a bit more comparable in size. And this one is, again, basically another plant. It came out 2 years ago, and from Japan, Kimuriella densifolia, and this is basically what the plant that Williamsonia and Welwitschia belong to. So this plant had, again, the same dichotomizing stems, but it seems like sometimes they didn't develop a side stem. They only developed one side. And the cones were born, again, in the center of these leaf clusters and then developed in the second year. And yeah, this is, the top right is, you can see the male organ. It was very deep. It had nectaries in the center and then pollen surrounding the outside. So it was lightly insect pollinated. And then we have the female cones, which again were quite narrow when developing, then expanded. They opened up at the top, allowing access for insects. And then in the second year, they would have developed into a cone. Yeah. Sorry, I'm having to go through this very quickly. Oh, I forgot about this one. Okay, so this is one of the bisexual taxa I was talking about, Williamsonia coronata. This one's a little different. So we have a potentially single specimen with corresponding leaf scars, which shows that, again, it dichotomized. But unlike the previous taxa, the cones were not born at the tip of a branch, but instead were born in the axils of leaves. Basically, they're born at the base of a leaf. So yeah, they would have formed really dense clusters of many cones in a single season, surrounded by leaves. The leaves were then dropped off in winter, leaving what seems to be very few actually pollinated cones. So we don't know how they were pollinated, but it has been suggested that several species of Jurassic flies pollinated them, which had really long proboscises. As you can see from the cone, we have the male outer part, which is yellow, surrounding the female inner cone. And these were all surrounded by 2 layers of bracts, which are probably just a single whorl continuing around. But the inner bracts were very delicate and probably light green or potentially coloured, because they have very few stomata. But the outer ones were very robust and dropped off very early in development, so are likely only there to protect the developing flower, or flower in quotes. Yeah, so this is probably the most important part for all the paleoartists. How would they have grown? Basically, we only have a couple of examples, but it seems like they would have formed these rather short, bush-like plants with canopies which basically obscured the underlying foliage. But there is diversity in growth forms even amongst what we know. So we have Wielandiella angustifolia up in the top, it's forming basically the standard plant. But ones with more compact upright branching, like Kimuriella, probably formed slightly more upright plants with a smaller canopy, with more branches exposed. And then Wielandiella villosa, which seems to be a lot more open, probably basically had most of the stems, or at least some of the stems, visible underneath the canopy. But basically, by altering only the divergence angle and the internode length, you can get all these different shapes of plants. There is one very unusual plant, which has not been described very well, which seems to show a sympodial trunk. Basically, the trunk grows, it divides into 2, but only one of them develops, and then that alternates, and it creates a single trunk. That could have produced a tree. But again, we do not know much about this particular specimen. It has not been described well. I would like to actually have a look at it to see what it shows. And finally, just want to talk a little bit about pollination. There does seem to be some diversity in pollination strategies within the Bennettitales. A lot of the Williamsonias are either very flower-like or more enclosed, so that could suggest that the large flower-like ones were pollinated by larger flying insects like the chromatids, which are a group of extinct lacewings which look like butterflies. The more enclosed ones may have been wind-pollinated or beetle-pollinated. We know that some Cycadioidea were beetle-pollinated, or at least facilitated. But there are a couple of male Welwitschias which seem basically suited to be pollinated by vertebrates. They were very long, conical, they had nectaries in the bottom, but they also had these recurved rays which would have prevented larger insects like calligromatids from pollinating them, and would have made it harder for smaller insects to also escape if they got in, so that's not useful for pollination. So they may have been pollinated by larger vertebrates, but we don't really know, because we don't have corresponding female organs which would have matched the shape, basically. So it's, at the moment, we only have one side of the puzzle. And yeah, that is basically it. I'm sorry that I had to rush through it. I ended up doing far too much for it. But yeah, if you want to screenshot this to read at your leisure, because I'm aware that we don't have much time. But yeah, basically these are the conclusions.
Yeah, you kind of get luck into questions.
Yeah, I'm sorry about that.
This is a good point to remind everyone that recordings of the talks will be available on Yasha's YouTube channel after the event. Yeah, I think we will have to cut the Q&A portion, sadly, but of course,
no, that's fine.
Q&A is available in the dedicated channel.